This is Dr. Daniel Yano from Falcon Physician Reviews. This section is Chapter 2, Section 1, Autonomic Nervous System. The motor or efferent nervous system has two components, the somatic and the autonomic. These two systems differ in a number of ways, but are chiefly distinguished by the types of effector organs that they innervate and the types of functions that they control. The somatic nervous system is a voluntary motor system under conscious control. Each of its pathways consists of a single motor neuron and the skeletal muscle fibers that it innervates. The cell body of the motor neuron is located in the central nervous system, or the CNS, in either the brain stem or spinal cord, and its axon synapses directly on skeletal muscle, the effector organ. The neurotransmitter acetylcholine is released from the presynaptic terminal of the motor neuron and activates nicotinic cholinergic receptors on the motor end plates of skeletal muscle. An action potential in the motor neuron causes an action potential in the muscle fiber, which causes the muscle to contract. For a complete description of the somatic nervous system, please see Chapter 1. The autonomic nervous system is an involuntary system that controls and modulates the functions primarily of visceral organs. Each pathway of the autonomic nervous system consists of two neurons, a preganglionic neuron and a postganglionic neuron. The cell body of each preganglionic neuron resides in the CNS. The axons of these preganglionic neurons synapse on the cell bodies of the postganglionic neurons in one of several ganglia located outside of the CNS. The axons of the postganglionic neurons then travel to the periphery, where they synapse on visceral effector organs such as the heart, the bronchioles, vascular smooth muscle, the GI tract, the bladder, and genitalia. All preganglionic neurons of the autonomic nervous system release acetylcholine. In contrast, postganglionic neurons may release either acetylcholine or norepinephrine, or in some cases, neuropeptides. Organization and general features of the autonomic nervous system. The autonomic nervous system has two major divisions, the sympathetic and the parasympathetic, which often complement each other in the regulation of organ function. A third division of the autonomic nervous system, the enteric nervous system, is located in a series of plexi within the gastrointestinal tract. The enteric nervous system will be discussed within the context of the GI tract in Chapter 8. The organization of the autonomic nervous system is described in Figure 2-1, which is also found on page 46 of your book, and there is a companion table, Table 2-1, found on page 47 of your book. The sympathetic and parasympathetic divisions are included, and for comparison, so is the somatic nervous system. Terminology. The terms sympathetic and parasympathetic are strictly anatomic terms and refer to the anatomic origin of preganglionic neurons in the CNS. For example, see Table 2-1. Preganglionic neurons in the sympathetic division originate in the thoracolumbar spinal cord. In contrast, preganglionic neurons in the parasympathetic division originate in the brainstem and in the sacral spinal cord. The terms adrenergic and cholinergic are used to describe neurons of either division according to which neurotransmitter they synthesize and release. Adrenergic neurons release norepinephrine. Receptors for norepinephrine on the effector organs are therefore called adrenoreceptors. Adrenoreceptors may be activated by norepinephrine, which is released by adrenergic neurons, or by epinephrine, which is secreted into the circulation by the adrenal medulla. Cholinergic neurons release acetylcholine. Receptors for acetylcholine are called cholinereceptor. A third term is called non-adrenergic, non-cholinergic, which describes some postganglionic parasympathetic neurons of the GI tract that release peptides, for example, substance P or other substances such as nitric oxide as their neurotransmitter, other than acetylcholine. Table 2-1 highlights some of the organizational features of the autonomic nervous system and contrasts them with the somatic nervous system as a way of contrasting the features of the broader efferent motor systems. Some of the major differences here are anatomic. So for example, the sympathetic division, as mentioned before, are thoracolumbar, whereas a parasympathetic have multiple cranial nerve segments, 3, 7, 9, and 10, and several sacral segments 2 through 4. There is a difference in the locations of the autonomic ganglia. The parasympathetics are primarily para and prevertebral, whereas in the parasympathetic divisions, these are found in or near the effector organs. The relative lengths of the pre- and postganglionic axons differ, such that the sympathetics have short preganglionics and long postganglionics, and vice versa in the parasympathetic nervous system. The effector organs are quite broad. The sympathetic and parasympathetic division broadly innervates smooth muscle, cardiac muscle, multiple glands, whereas the somatic nervous system innervates only skeletal muscle. There are some differences in the neurotransmitters and some similarities in terms of the ganglionic neurotransmitters. Both the sympathetic and parasympathetics use acetylcholine and nicotinic receptors, and at the effector organ, the sympathetic division uses various forms of adrenoreceptors, alpha 1, alpha 2, beta 1, or beta 2, whereas the parasympathetic uses primarily muscarinic receptors, and the somatic nervous system uses acetylcholine and nicotinic receptors. To summarize, whether located in the sympathetic or in the parasympathetic division, all preganglionic neurons release acetylcholine and are therefore cholinergic. Postganglionic neurons may be either adrenergic, meaning they release norepinephrine, or cholinergic, meaning they release acetylcholine. Most postganglionic parasympathetic neurons are cholinergic. Postganglionic sympathetic neurons may be either adrenergic or cholinergic. Most of them are adrenergic, but there are certainly some cholinergic neurons in this class as well. Figure 2-1 illustrates some of the organizational principles of the various nervous systems. As mentioned before, in the somatic nervous system, this is a single neuron pathway from the CNS out to the effector organ, whereas in the autonomic systems, these are two neuron pathways. In the sympathetic, we have a short preganglionic cholinergic neuron synapsing on a nicotinic receptor, and then a long postganglionic, typically adrenergic neuron, although there are cholinergic neurons as well. The adrenergic neurons use some form of adrenoreceptor, alpha 1, alpha 2, beta 1, or beta 2, whereas the cholinergic neurons are muscarinic. The parasympathetics have long preganglionics, short postganglionics, again acetylcholine at a nicotinic receptor at the ganglia, and acetylcholine at a muscarinic receptor at the effector organ. And then finally the adrenal medulla, which is essentially part of the sympathetic nervous system, has a short preganglionic, and there is no postganglionic axon. The postganglionic cell is a chromaffin cell that releases primarily epinephrine, approximately 80%, and less norepinephrine, approximately 20%, directly into the circulation. Neuroeffector junctions of the autonomic nervous system. The junctions between postganglionic autonomic neurons and their effectors, or their target tissues, the neuroeffector junctions, are analogous to the neuromuscular junctions of the somatic nervous system. There are, however, several structural and functional differences with the neuromuscular junction. Number one, the neuromuscular junction, as discussed in Chapter 1, has a discrete arrangement whereby the effector, a skeletal muscle fiber, is innervated by a single motor neuron. In contrast, the autonomic nervous system, the postganglionic neurons that innervate target tissues form diffuse or branching networks. Beads, or often called varicosities, line these branches and are the sites of neurotransmitter synthesis, storage, and release. Varicosities are therefore analogous to the presynaptic nerve terminals of the neuromuscular junction. Number two, there is overlap in the branching networks from different postganglionic neurons, such that target tissues may be innervated by many postganglionic neurons. And then number three, in the autonomic nervous system, postsynaptic receptors are widely distributed on the target tissues, and there is no specialized region of receptors analogous to the motor end plate of skeletal muscle. That concludes Section 1 of Chapter 2.